Joining me today is Professor Hans-Georg Rammensee, the head of the Department of Immunology from the University of Tübingen. Thank you for joining us today, Professor Rammensee.
Hello, thank you for your interest.
Now, a key area of your research is therapeutic vaccines in oncology. For everyone not familiar with the subject, could you perhaps briefly recap what this is about and what therapeutic vaccines are?
Well, they don't exist so far, at least not as successful vaccines. But what is clear is that the immune system can fight against cancer, and the best example is so-called checkpoint inhibition, and this is shown here in this picture. Checkpoint inhibition means that tumor-specific T cell, which exists in some patient, is activated by specific antibodies, which can be considered as antibodies losing a break, and then these T cells can kill the tumor cell, which is down here. And these are the discoverers of these checkpoint inhibitions who received the Nobel Prize, Tasuku Honjo and Jim Allison. And the result of their discovery is that these breaks can be removed in the interaction between tumor-specific T cell and tumor, which then can lead to the resolvement of the tumor, so the patient gets rid of his tumor. And this was a great success in cancer therapy in general, and was essentially the beginning of real cancer immunotherapy. Problem is, this kind of cancer immunotherapy with a checkpoint blockade, as it is called, works for some cancers, like for melanoma and for some others, but even there, not for all patients, only for, let's say, a third of the patients, or maybe a little bit more now. But for many frequent cancers like colon cancer or breast cancer, this checkpoint inhibition does not work, essentially not work. And so there is a question, what should one do with the majority of cancer patients not benefiting from checkpoint inhibition or checkpoint inhibition alone? There are several reasons that they do not respond. And the key to this question is the interaction between the T cell and the tumor cell, where this roundish structure peptide is at the center. So this interaction works that the T cell receptor recognizes a peptide, a small molecule, a protein fragment on the MHC or HLA molecule of the tumor cell. And if the T cell receptor fits together with this combination of antigenic peptide and MHC, and there are some other conditions like no break and other activating steps, if they are fulfilled, then the T cell can kill the tumor cell. So what we have here at the center is a peptide sitting on the tumor cell, which is tumor specific. So there is reasoning that maybe therapeutic vaccination is one option for the majority of the cancer patients not benefiting so far from checkpoint inhibition alone, because therapeutic vaccination should induce, I go back for one, should induce T cells against such peptides, even if they are not induced by the presence of the tumor alone. There are several attempts have been several attempts for therapeutic vaccination, like these two from Mainz, Germany, and from Boston, Massachusetts. They both used so-called mutated antigens or neoantigens, which are these peptides in a mutated version, differentiating it from between tumor cells and normal cells. But the problem is that in checkpoint blockade, these T cells recognize, the induced T cells recognize mutant antigens, mutant peptides. The problem is that most of the mutations found at the DNA level do not turn up as HLA presented peptide or MHC presented peptide, which means one can predict such mutated peptides from from sequencing results, but in most cases, these peptides will not be presented by HLA on the tumor cell for a number of reasons, and might be different by cross-presentation and the so-called MHC class II molecules. But by and large, the mutated neoantigens appear to be very rare in many tumors. So one cannot take these mutated antigens as tumor-specific antigens. But we know that there are many germline sequence tumor-associated and also tumor-specific HLA peptides, presented peptides, and there are T-cell responses against these non-mutated peptides. And our approach is to find such peptides. Now one has to say that so far there was no real successful therapeutic vaccination, at least up to phase 3 trials, neither with peptides or anything else. But there is a large number of earlier trials or case reports indicating that T cells can be induced against cancer and they can sometimes have benefit. But the general problem with most attempts of therapeutic vaccination, in particular with peptides, but also with other substances like RNA or proteins or virus vectors, usually there is a weak immune response and that's combined with strong adjuvants. That's why our strategy in our department is here in Department of Immunology, in collaboration with the university hospital, is first to identify the relevant tumor antigens, and it doesn't matter whether they are mutated or not. And the second, I will come later on to this, induce efficient immune response by strongly adjuvanted vaccination. So our approach is we try to identify tumor-specific peptides among all the HLA ligands on the tumor. And these are peptides. The definition of our tumor-specific characteristic here is peptides is present on cancer cell but absent on normal cells. This is our standard, our method which we developed many years ago to isolate naturally presented HLA ligands from tissues, for example, cancer tissue or normal tissue. We produce tumor cell tissue lysate, analyze and purify the MHC molecule. MHC and HLA is in this case essentially the same, and then elute the bound peptides from HLA and identify them. And here you see this is an HLA molecule. This little thing is a peptide in it, and the hand you will see now is an antibody precipitating the HLA molecule, and we take out the peptide, collect it. These are actually, it's not one peptide, but thousands of peptides. These peptides are then separated by HPLC and analyzed by mass spec. And in the meantime, in the last 10 or 15 years, we have done this for a large number of tumor samples and also a large number of normal tissue samples. And we have now a database of several million of such peptides found from tumor and normal tissue, which is very large in the meantime. It's also published in a preprint server. I will come to this. Important here is that in every human, there is a different HLA, a set of HLA molecules. So this wooden model shows an HLA molecule, a frequent one, HLA-A2. Little thing is a peptide presented by this HLA molecule, and this thing on the top is a T-cell receptor. And the picture here from the well-known TV family should just indicate that every person, even within a family, has a different set of HLA molecules, and this different set of these different HLA molecules present different peptides according to certain rules, which we cannot go into now. But you see from these colored pictures, each of the HLA molecule has a name, a number, a letter and a number, and each of these molecules have different peptide specificities. So in consequence, every human being presents a different selection of peptides from a given protein. This actually also might explain the different sensitivity of people to infectious viruses like the coronavirus. And we have published this in the preprint servers so far. So the Hla Ligand Atlas, which is essentially just a list of the HLA ligands we find on normal tissue. I will briefly just go over it, just to indicate there is awful lot of data in it, many peptides. Thus, we know a good deal of the Hla ligands present by normal tissues, and this enables us to judge the tumor specificity of new peptides we find on tumors. And now we have done this for several tumor entities, leukemias, it's all published, and actually we are running a leukemia a peptide vaccination study in leukemia, which is has this number in CT something, and a new version of this trial is in preparation together with our new adjuvant, which I will come later to. And then also ovarian carcinoma, we had lots of data, also colorectal cancer, many others, and also brain tumor. And based on this analysis of brain tumor, glioma peptides, we have in a large European consortium, in so-called GAPAC consortium, we managed to do an actively personalized vaccination trial for glioblastoma, where we use, this is a complicated setting, but the whole GAPAC trial was complicated. So what we do is we analyze the tumor of the patient, the brain tumor, by mass spectrometry, look for peptides which are tumor specific, and we selected peptides we knew before from other patients. And for each of the patient, 16 altogether, we put together tumor specific peptides which are not mutated but tumor specific. And this was formulated in a vaccine cocktail we call APVAC1. These are non-mutated peptides from a pre-manufactured warehouse, which could be produced rather rapidly. And during this time of first couple of vaccinations, mutated peptides were looked for based on gene sequencing and prediction and mass spectrometry. And the second cocktail, APVAC2, should have contained mutated peptides validated by mass spec, which we could not fulfill, but we used predicted mutated peptides and vaccinated these patients with two different peptide formulations then. And this is the schedule. This is published in Nature last year. But important message here is that both the non-mutated tumor-specific peptides as well as the predicted mutated peptides gave an immune response, class 1 and class 2, so killer cells and helper cells, by and large in a similar strength. So one can use non-mutated tumor-specific peptides to induce T-cell responses in patients, and these T-cells actually, we have seen in one case, go to the tumor. In this case, the patient had a recidive, a relapse of the tumor, and we could find such vaccine-induced T-cells in the recurrent tumor. So this study did not show clinical benefit. It was not designed to this, but it could show, this study did show that we can induce T cells, and especially that we solve the regulatory issues with such a personalized vaccine schedule. We can do such things because we have this GMP Center for Individualized Substances here at our university and at the University Hospital, where we can produce. This is our document showing that we have the drug production approval for this purpose from the local government, from the government authority. And this is a complicated process, so just to show that it's a lot of documents and a lot of clean room handling. So we manage this, and that's why we can do such individualized cancer treatments. Now a problem is, so this part so far should have shown that we are able to find the right peptides, mutated, non-mutated, which are tumor specific. And now what is not solved so far is to induce a strong T-cell response against such peptides. Because this is, as I mentioned before, the general problem with therapeutic vaccination so far, only weak immune responses are induced, and that's combined with strong adjuvants, but there is no really good strong adjuvants for this purpose. So that's the second part here of our strategy. We try to induce efficient immune responses by strongly adjuvanted vaccination. And for this, many years ago in 1989, we showed in a mouse that there is this substance from bacteria, pemphigus, induces strong T-cell responses in the mouse. And based on this, we designed a new molecule, which is shown here, pemphigus with a peptide on it, which we call XS15, just to make it shorter. That's its structure. And it induces a strong T-cell response in men. We tried this first with these 3 viral peptides. So the peptides are mixed with adjuvants, XS15, and also with montanide, which is an oily substance, essentially the old incomplete Franz adjuvants. And with this, actually, I tested this in myself to make it faster. We saw strong T-cell induction. So these spots here show immune response before vaccination, and this down here after vaccination. And each spot is a T-cell colony. So the more spots, the stronger was the T-cell response. And there was a local reaction which could be seen at the abdominal injection site, indicating that there is an immune response going on. And even in the injection site, in the granuloma, we saw functional active T cells. We also found the peptides again by mass spec analysis after several weeks of the injection. And we found in the granuloma, which was cut out just to analyze it, we found T cells, CD4 and CD8 T cells, macrophages, B cells, and still the peptides left, and lymphoid structures. And we think this is why this approach works so well, because we get a new formation of lymphoid structures at the injection site. And this kind of vaccination induces strong T-cell responses. This is a different experiment. Is again shown that it uses strong T-cell response. And important here is that that such T-cell induction lasts for a long time, more than 1 year. So this is adjuvants used in the peptide used in the first vaccination, and more than 1 year later, a second vaccination was done with the same peptide used here as a control. The T-cell response from the first vaccination after more than 1 year was still left as a strong T-cell response. It could be very strongly boosted, as you see on the last dots. So we can induce, this is a summary, we can induce strong T-cell responses against peptides, and this works not only for viral peptides as shown before, but also for mutated and for non-mutated self-peptides. We have shown this in a number of individual treatment cases in patients, and we have published this adjuvants in recently, well, 1 year ago. And we hope that this year, maybe next month already, October, we can have the first real clinical trial with these new adjuvants.
Thank you very much for these really interesting insights in your research work, Professor Rammensee, and thank you very much for taking the time to talk with us today.
You're welcome. It was fun. Yes, thank you.
Thank you.